This recorded session is dedicated for the completion of module number 5 on microbial habitats. So during our last synchronous meeting, we were able to discuss about the variety of habitats where microorganisms are expected to survive. No? So we've talked about aquatic, terrestrial ecosystems. So under terrestrial ecosystems, we've mentioned um, the soil as a habitat, rocks, and then underground or subterrestrial habitats like caves and groundwater we've also discussed. In aquatic ecosystems, we've mentioned about or we've talked about marine water and freshwater habitats. Then for atmospheric habitats, we've made a quick review on the atmosphere as a transitional matrix, no, allowing microbes to be transported from an original place, from their source to another location. Now, to complete our discussion, we're going to proceed to the final topics. So we start with population ecology across different habitats. If you would remember from our first ever synchronous lesson or synchronous session, we've defined what microbial ecology is all about. And in this discipline, no, one of our major objectives is to be able to explain distribution as well as diversity of microorganisms by looking at how they work or looking at how they do their cellular processes, their physiology, how they interact with other organisms, other microorganisms or macroorganisms within the habitat that they are living in. And then also um, the state of the physical chemical parameters of the environment, that could also serve as a independent factor affecting the distribution and diversity of microbial communities, microbial populations. And of course, I cannot emphasize in I cannot emphasize anymore na very important concept that we're really always reverting back to is genetic diversity. Genetic changes at the level of the cell, of the microbial cell, or at the level of the population in response to environmental factors. Although this has been, a major challenge in studying microbes, especially those microbes in aquatic environments, very important that we're considering such independent factors whenever we really want to focus on discovering diversity distribution patterns of our microbial organisms in their specific natural habitats. No? So, in archaea and bacteria, to be particular, we have the concept of ecotype. Though actually, it's not the concept of ecotype is something that is not confined to prokaryotes, but for archaeans and eubacteria, this is very the phenomenon or the the process of the emergence of several ecotypes of a specific species is actually very fast compared to ecotypes in as used in macroorganisms, no? Kasi nga, diba, a lot of changes or a lot of or essentially the adaptation of a microbial organism to its environment is really genetic in nature. And um, the interaction or the total effect of how the microbe really selectively adapt or really adapts to the environment and the environment selectively or exert selective pressure doon sa mga nagso-survive sa kanyang living entities, living systems, no? So in bacteria, the production of an ecotype is faster. The rate of ecotype diversification is faster, no? Kasi there are other factors, no, aside from genetics itself, the genetics of the bacteria. Other factors such as ultraviolet radiation, which we know can cause mutations in the bacterial, in the prokaryotic genome, errors in DNA replication. In some instances, gene duplication also serves as um, an additional factor affecting microbial genome diversity. Then we also have horizontal gene transfer, which we will talk about in a little while. Gene loss is also another thing. Though gene loss or reduction, gene reduction, genomic reduction is something na hindi common among prokaryotes, but as an event, it still contributes to variation of microbial genomes. And then lastly, we have homologous DNA recombination, which is the usual process ng the usual process during bacterial division. We expect homologous DNA recombination, no? Okay, so um, aside from the idea that in microbes, population ecology across habitats is really defined heavily by how the microbe will respond, how its genetics will be able to adapt to the selective pressures of the environment, no? One additional complication, that's why we're really having a tough time in understanding or generalizing microbial ecologies of different habitats, is the fact that until now, the idea of a prokaryotic or microbial species is really difficult to articulate, or it's really difficult to, standardize. No? And with such, no, if we are really trying to uncover the diversity, the microbial diversity of an environment, it will take more than one, let's say, marker gene to really study or to really capture the microbial diversity of a specific environment. To some extent, ngayon, no, parang gumagamit na rin tayo ng whole genome, or there are various attempts on capturing microbial diversity of a natural habitat, not by using one marker gene like 16S, but the whole genome of all organisms present in the habitat. No? So now we're going to proceed to give special emphasis on horizontal gene transfer in a little while. But first, let's have some discussion points on population growth and dynamics of microbes. No? So let's remind ourselves that the amount of population growth by microorganisms in across the habitats, in different habitats, is governed by by a lot of factors like resource availability. Under resource availability, meron bang essential macronutrients that are present. Then also, are there chemical compounds that can serve as electron acceptors and electron donors. So all these resource categories, no, are influencing heavily how a bacterial species or a bacterial community will continue growing and will continue interacting with one another. Um, in additions, also, there, aside from resources, no, resource availability, other abiotic factors are also factored in whenever we want to capture population growth and dynamics. This includes seasons, temperature, niche space, as well as what they call this, microbe interactions, no? Okay, so those other, actually, sorry, not really microbe interactions, because hindi naman siya categorized as abiotic factor, pero yun, the seasonality, or the fact that habitats experience seasons, the fact that temperature differs all throughout the day, and then the microorganism will occupy a particular niche within the habitat. All those things, added with resource availability, will actually affect how a microbial species or a microbial community will continue to grow. No? Now, okay, so um, one successful trait of dominant microbial communities, microbial populations, or species in a particular habitat is the fact that they can enrich their genome. The fact that they can, through time, they will be able to diversify and increase their genomic features or genomic capacities. No? And a key activity or a key phenomenon in prokaryotes is horizontal gene transfer, so HGT. Now, not all genes are transmitted vertically during, prokaryotic division. There is substantial evidence that that some specific genes, especially those that are implicated in pathogenicity, okay, are actually transferred horizontally or laterally, meaning the transfer of the gene is not between a mother cell to a progeny cell, but between two cells coexisting at the same time. No? So in bacteria and in archaea, horizontal gene transfer occurs by various or through various mechanisms. No, so probably you are already aware of these activities or these processes. So HGT can occur through transformation, through transduction, or conjugation. Diba? Now, it's widely accepted that HGT is expected to occur at higher frequencies between relatively similar bacterial strains. No, however, that's not really a general rule because there are instances in which, or there are observations in which bacteria and archaea actually shared or there certain bacteria acquired archaeal genes. No, also as a good example of that, no, we have um, here we have Aquifex aeolocus and Thermotoga maritima. They have been documented to contain many archaeal genes, no, probably because they acquired it from, the thermophilic archaea that share the environment with them. Then aside from that, we also have in microbes that undergo photosynthesis. Now, there's good evidence existing for the potential role of horizontal gene transfer in the transfer of genes important for photosynthesis by cyanophages. No, which actually act as a gene with yung ano no organism, no, is capable of being the vector also of such photosynthesis genes na pwede siyang mai-transfer to a bigger microbe probably, and that microbe become capable of photosynthesis as well. No. So, however, even though pala, no, we have growing evidence on the role of horizontal gene transfer in prokaryotic diversification, in microbial community, the enhancement of the genomics, no, or metagenomics of microbial communities, there are still opponents, or there are individuals who oppose the idea, and as an argument, no, they use the idea of evolutionary convergence. Kung saan daw it's possible na um, in instances where a prokaryote A also possess the similar genes with prokaryote B, instead of, HGT as a plausible explanation, it could be evolutionary convergence, because if, let's say, prokaryote A and prokaryote B, they live in the same environment, the same natural environment for the longest time, so they're experiencing the same conditions all the time. So it's possible that the effectors like proteins, no, are products of evolutionary convergence and not really transfer of genetic elements in a lateral or horizontal fashion. So overall, I mean, on your own, I will leave it to you, whatever you want to, cling on to. But these two things are plausible hypotheses, and perhaps, what we can be reminded about, or what we can reminded of is, maaring may mga dedicated conditions needed in order for them to really occur. And I think for the idea of evolutionary convergence, time is a big factor. Time, it will really take a lot of time, I guess. Even for prokaryotes, no, I think it will take a substantial amount of time if we are to explain such a phenomenon with evolutionary convergence idea. Now, overall, even though HGT is not really applicable all the time, or it's not really there's no observable evidence all the time that it's really working. No, we can say that as a process, it's a very important evolutionary mechanism, no, or life mechanism of bacteria and archaea, such that they can increase their genetic diversity, and they can adapt to a wider range of environmental conditions, or to a widening range of environmental conditions, as we see na mas marami pang factors actually disturbing the usual parameters of a specific natural habitat. Okay. So now we're going to talk about the last topic for module number 5, which is biogeography, no? Biogeography and the idea of cosmopolitan organisms. So I think, or I'm in our laboratory session yesterday, or last Wednesday, we talked about the concept of cosmopolitan organisms or cosmopolitan microbes, especially in aquatic habitats, no? Yung sinasabi nga na everything is everywhere but the environment selects, no? So actually the idea of cosmopolitan organisms or cosmopolitan microbes is in contrary to the concept of biogeography. So biogeography is where is actually the refers to the study of the distribution of species and organisms in ecosystems based on geographic space through geological time. So, the idea of biogeography is, or biogeography posits that there are geography induced differences in terms of organismal diversity. Ibig sabihin, the microbes that we see in the Philippines are expected to be different from the microbes that we can observe in other parts of the world. Okay? So, generally opposing sila, and on its own, no, biogeography is really very much well documented, no? Kasi we know for a fact that organisms and biological communities will definitely be affected by geographic gradients, elevation, isolation, and the habitats, natural habitats in different continents or different geographies are expected to be of different nature then, no? So uh, that's one idea, yung diversity because of geography, because of time and isolation, geographic isolation. Now, for certain natural habitats naman, it's easier, or it's more, I would say that mas heavy yung evidence to believe that there are cosmopolitan microbes, and even at the taxonomic level, we can identify really we can really identify microbial groups that can exist in a very wide range of parameters, such as Flavobacteria, Actinobacteria, the phototrophs, the cyanobacteria, and the proteobacteria, especially beta proteobacteria. These cosmopolitan microorganisms, they have high dispersal rate. They can be distributed across continents, and then normally, they they don't have spatial clustering within the different locations of the planet. So we can really say na cosmopolitan sila. Now, um, the thing with cosmopolitan microorganisms is that yun nga, if there are groups that can really survive in the all the ranges of physical, chemical, biological parameters in all types of habitats, no, what really the biggest question na we can really get, or the biggest question that will arise from this discussion is, how does the environment, or what makes such cosmopolitan microbes survive in a particular habitat na, let's say for example, has become so specialized that it no longer possesses a generalizable physical and chemical a generalizable range for its physical and chemical characteristics. So that's yun nga yung idea na it's really the environment, no, that eventually will finally select what microbes or microbial communities it will allow to survive within its, within its vicinity, no? So in um even though, no, but even though the schools of thought ng biogeography and environmental selection very different, they can be unified by several microbial processes such as dispersal, speciation, and then extinction. No, so those no, biological phenomenon that microbial communities, microbial species are doing or are experiencing from time to time. Those are unified activities or similar activities that we can, factor in whenever we want to discuss microbial diversity distribution, either using the school of thought of biogeography or the school of thought of cosmopolitanism, na only the environment, once settled yung mga microbes, will now start the picking or selection process. Okay? So, as a summary, no, we talked about how current research is moving towards a direction of of a direction of understanding or defining what microbes are present in a specific habitat, to to the idea or to the question of what controls the presence or the diversity of such microbes in different habitats. So I think there's really global movement or global, transition from the fact na, or from the initial energy na we want to know what microbes are present in a specific habitat, to knowing what factors, what parameters really decide on the dominance or the recessiveness of a specific microbial species, population, or community in a given habitat. No? So let's remind ourselves that, such parameters, resource availability, the physical chemical parameters, they are really at a certain instance, probably, during the earliest point in the development of our planet, pare-parehas lang yung status ng mga yan. Pero as we proceeded with, through time, we expect, we can conclude that specializations within habitats are now existing. No? So um, now, I want you to take a look at the synthesis questions I've placed here, and on your own, no, I want you to try to answer them, or try to reflect if you can really answer them, since we've accomplished our lecture discussion already. No? And this will really help you in preparing for our summative assessment. Okay, so that's the end of our module number 5 on microbial habitats.